Hello friends, welcome back to the journey of microbiology. Till now we start discussing about the chapters called the immunity, and under this chapter we discussed about the immunology, a branch of infectiology in which we studied about the entire aspect of immune system. Then we studied about that immune system is classified into the two groups based on their origination. First we described already about the innate immunity, also called inborn immunity, also called as non-specific type of immunity, and we also studied the barriers, the four different types of barriers. Okay, then we move ahead and then we studied about the acquired immunity, also called as adaptive immunity, also called as specific immunity. Clear? That we studied that this acquired immunity is mainly generated or mainly done by the help of lymphocytes, which is basically a granulocyte, a type of leukocyte that is called the WBC. Okay, WBC is a one cell component. Okay, and this cell component is divided into the two parts, granulocyte and agranulocytes. Okay, under the agranulocyte these lymphocytes are come, and these lymphocytes are the cells which help in the acquired immunity and adaptive immunity. Okay, then we studied that there are two types of acquired immunity, or we can say the lymphocytes. Okay, this lymphocyte, first one is called the T lymphocyte and second one is called the B lymphocyte. Okay, T lymphocyte is responsible for the cellular immunity. T lymphocyte is involved in the cellular immunity, and B lymphocyte is involved in the humoral immunity. Okay, then we studied that the T cell, which is produced from the where it's produced from the bone marrow mainly. Okay, but we looked depth and we seen that during the fertilization, when the sperm and egg they fertilize, okay, they form the zygote, and that zygote undergo the subsequential division, okay, and differentiations. They produce the embryo, and that embryo, there is some of the hemopoietic cells, which is like a stem cell. Okay, and these stem cells eventually transfer to premature fetus liver. Okay, and those premature liver cells again transferred to the where bone marrow. Very good. And in bone marrow those cells are known as the progenitor cell or pluripotent stem cell. Those cells are known as the pluripotent stem cell, and in that pluripotent cells which is present in the bone marrow, they divided into the two types. Okay, the first is called the lymphoid progenitor cell and second is called the myeloid progenitor cells. The lymphoid progenitor cells, they further, we can say, differentiate into the different types of cells. Okay, so now we are doing first seeing the slide. Okay, because already we discussed. Now we see the slide, quickly recap and then move ahead to understand about some of the more topic in immunity. Okay, so see the slide first. We studied about the production, or we can say the differentiation or maturation of T cells, which is done mainly in the thymus and T cell development. This T cell thymus development is also called T cell lymphopoiesis. Write down this word, T cell lymphopoiesis. T cell lymphopoiesis. Always remember, this poiesis means production. This poiesis word denotes production. Clear? And here which production? T cell productions. But this T cell lymphopoiesis describe all their production as well as their, we can say, maturation. Clear? These two things is done by the T cell lymphopoiesis. Okay, now see the slide. We already discussed that in the bone marrow, there is something present called the pluripotent stem cell. There is a presence of pluripotent stem cells, and these pluripotent stem cells, they divided into the two types. Okay, they divided into the two types. The first type is called as the lymphoid progenitor cell, and second type is called as the myeloid progenitor cells. These lymphoid progenitor cells, they are divided into the three types. First one is called B cell progenitor cell, second one is called as the natural killer cell, and third one is called the T cell progenitor cells. Okay, these T cells, they goes where? To the thymus. They goes to the thymus, and these start under the effect of thymosin, thymopoietin and thymotactin. We already described about these three types of secretions coming from the where? Coming from the cortical area of thymus gland, coming from the cortical area of thymus gland. In the cortical area of thymus gland, there is the presence of epitheliocyte. Okay, and this epitheliocyte secrete three substances, namely thymopoietin, thymotactin and thymosin. This thymotactin is involved in attracting the premature T cells from the blood, from the circulation, because when the bone marrow releases the newly formed T cells, they first went into the circulation, our blood circulations, and then due to the involvement of these secretions, mainly the thymotactin, they attract those T cells and trap in the thymus outer area. That's called the what? That area is called the cortical area, or basically the cortex. Okay, in the cortex there is a start activation, or we can say the specialization, proliferation or differentiation of those newly T cells. Okay, and eventually they develop one TCR, that's called the T cell receptor, and these T cell receptors, they have highly important. They are the highly important receptor, because by these receptors T cell recognize the foreign antigen. Also they differentiate on native antigen. Okay, so that they don't destroy the other cells. They don't work blindly as our specific immune system is working. Sorry, our non-specific immune system, that's called the innate immune system, is working. Clear? Now see the second diagram. Here there is a beautifully described that when the bone marrow sended their newly formed T cells into the cortical area of these, we can say the thymus gland, here due to the effect of those thymosin, thymotactin and thymopoietin, T cell differentiated into the four type of cells by the processing of T lymphocyte, Okay, we already discussed that there are 5 events in which these T cells are differentiated. Okay, they differentiated into the 4 types, and just I am remembering you the names. The first cell we discussed is called as the cytotoxic cell. Okay, or also known as the CD8 cells. Another cells we have, apart from the cytotoxic CD8 cells, we have the another class of cells, and that another class of cell is called as CD4. Okay, CD4 cell. Then we have memory cell. And at last we have the another kind of cell, and that another kind of cell is called as, that another type of cell is called as simply, that another kind of cell is called as the memory cells. We already described that memory cell and the CD4 cells, they are also these cells are also known as the helper T cell. They are known as the helper T cell. Why this helper T cell I will describe. Okay, and last one is our have the suppressor cell. Suppressor cell. See, these CD8 molecules and CD4 molecules, okay, these are main, we can say, the T cell differentiation. Okay, these are the two main T cell differentiated cells, okay, based on the charge, selection of the charge, positive charge selections, they are both classified. Now, how you will remember these names? First, you think that why they are called helper T cell? There must be any reason, underlying reason. That's why they are called the helper T cell. See, you know, we have two hands. We have two hands and two legs. Okay, we can help someone by our both the hands and both the leg. Okay, so you just remember by this one technique that CD4 means helper cells. Remember, two hands, two leg. It means it's a helper cell. Whereas cytotoxic cells, you know, if you help someone at some extent, that's wonderful, okay, that's a welfare for someone, but if you help too much to someone, it's become toxic to you. That's why you just remember by this story that cytotoxic cells means CD8 cells. You are helping too much, you by your 8 hand, 2 hands, 2 leg, and 2 imaginary hand and 2 imaginary leg. Okay, clear? Now we have the memory cell and another one is called the suppressor cells. Okay, they are further differentiated based on the antigen interaction. Based on the antigen interactions, they are further classified. Okay, so now we are moving ahead and understanding the function of T lymphocyte. Okay, we are moving ahead and understanding the function of, hope you clear with these things. Okay, now immunity is something very useful for us. Suppose if we don't have the immune power, then what will happen? We will suffer a lot. We will suffer a lot because we are living in that world which is too much fast. Okay, they are not taking care the things properly. Why I am saying properly? Because we are exposed. We are not living in our ancestral type of life. Okay, we have advanced things. Okay, and that advanced thing generating so many very deadly kind of substances we call as the foreign particles. You know, the factory exhausted, the factory smokes. Okay, there are so many different kind of allergens, okay, which arises from inhaling, injecting or exposing to the environment. Okay, so just because of this, our immune power, immune system, our rest of the system is protected. Even our nervous system is also protected from by the immune system. Clear? Now we are moving ahead and understanding the function of T cells. Function of T cell. T cell function. Now you know that primarily they are involved in destroying the microorganism. They are primarily involved in the destroying the microorganism. Suppose, take an example. See, this is our one beautiful T cell. I must say it beautiful because they are very precious. They are very important, useful for us. Okay, and they have nucleus. Without nucleus, a cell cannot considered as alive. No, except RBC. Very good. A mature RBC doesn't contain the nucleus. Okay, there is one story we will discuss later in hematology. Okay, now understand these are the receptors, okay, which developed. These are the receptors which developed during the processing in the thymus gland. Okay, now you have, suppose we are affected by one bacteria. Suppose this is one diagrammatic representation of bacteria. Bacteria have the cell wall along with the cell membrane and their nucleus. Okay, now it's not have a nucleus because they are prokaryotes. They have the plasmids, again, and other structures. Okay, understood. Bacteria secreting their antigen. Bacteria secreting their antigen. Okay, by the means of, we can say, the endotoxin and exotoxin. Endotoxin and exotoxin, they are the toxins. Okay, so collectively we are just understanding they are the toxins. They are releasing the toxins, and these toxins enters in our cellular body compartment, okay, and trying to replicate, enhancing their progeny. Okay, they are not doing anything bad, they are doing best for their survival. Understood? They are doing best for their survival, because you know, as a human beings, we have also the tendency, we eating, okay, sleeping, taking the rest, and enhancing our progeny, leaving the we can say the print for our future. Okay, that's basically called the reproduction. So they are doing the reproduction, so they are secreting some of the toxins, and these toxins try to start residing, or we call this adhesion. Okay, they are trying to adhesion on our tissues, cell surfaces. Okay, but our cells have the immune cells. Our cells have the immune cells, and those immune cells, they won't allow any other kind of toxins to come, because their toxins secrete antigens. Okay, and these antigens, they are not a native for us, means they are not a local, it's not produced by our body. So our body have some of the immune cell which have the capability to recognize the foreign particles. So these are the foreign particles. Clear? Now these foreign particles, they try to adhere, they try to make attachment or adherence on our tissue surfaces, body surfaces. Okay, but before coming to where the internal environment, cellular environment, these toxins have to fight with our very good innate immunity, which providing the different kind of barrier, like a physical barrier, physiological barrier, cellular barrier. Okay, some of the cytokinins, if they will potential enough, if they will strong enough to enter inside those all barriers, then they come into the cellular environment. Okay, now our cellular environment have the immune cell. Clear? These immune cell are doing what? They are sleeping cells. Even though there are plenty of amount already stored, I told you, into the lymphoid organs. They are numerous. They are not a 1, 2 or 100. They are in the thousands. But they are in a sleeping mode. They are not activated till now. Okay, we are not calling it a activated T cell. They are simply now a T cell, or we can say the naive cell. Okay, newly formed cells. What happened when these foreign particles, toxins enters, they make the adherence, they disrupt all those kind of four innate barriers, we can say the barrier, they come into the cellular environment, they bind, these toxins bind with one of the most important molecules in our body. Can you guess which kind of molecules? Do you heard about tissue histocompatibility, organ transplantations? Okay, these are the term related with the matching process. These are the term, I am simply describing it. These are the term related with the matching process. Okay, first we go slowly. MHC molecules, they bind with the MHC molecules. Now leave this process in mid and first understood about the MHC molecules. First understood the full name of these MHC molecules. These MHC molecules is called as major histocompatibility complex. write down the word measure histocompatibility complex. Now you are thinking what this is new thing is. There are very new things in our body. Till now lots of things is discovered, till now lots of things is pending, but that will be revealed in future. Okay, so that's called the major histocompatibility complex. This complex, they are doing so many things. Okay, they are doing so many things, but their fundamental work is they are the one who can differentiate. You know, I already told you in the previous lectures that immune system is something which is masters in discriminating or differentiating between own cells and different cells. Okay, but in a depth way, these are those molecules which are doing this kind of job, differentiating. Understood? Now these MSC molecules, I am going bit detail, but we will cover this topic again in our further subsequent lecture. But for right now, for understanding the functions, we have to understood these MSC molecules. So the MSC molecules, also called major histocompatibility complex. Major histocompatibility complex. This complex is the one who is differentiating. They are the differentiator cells, differentiator molecules basically. They are not a cell, sorry. They are a differentiator cells. See, this is produced by the chromosome number 6. We have chromosome, how many chromosomes we have? 46 chromosome or 23 pairs of chromosomes. In this 23 pairs of the chromosome number 6, chromosome number 6 is the one gene which involved in the synthesis of this protein. Basically they are the protein or molecules, MSC molecules, major histocompatibility complex. Now I will not go today detail about the MSC molecules because they are diverse, just understood some of the points. These MSC molecules, they are involved in the organ transplantation. Suppose I have one failure, okay, not a live failure, but suppose I have any organ failure. Take an example for the liver. Okay, I have the liver failure, then I am searching one donor. Okay, then you know there is something called matching, tissue matching. If tissue matching will not happen properly, then there is something called the organ rejection or tissue rejection happen. Okay, in that matching, this MSC molecules matching is really important thing. If these MSC molecules are not matched, okay, our cell will discard those newly transplanted organ. For example, liver, they won't allow to adjust, or we can say in a simple term, fit. Okay, so in nutshell we can conclude that MSC is the one molecules which produce from the chromosome number 6 by the involvement of chromosome number 6, which differentiate the organ transplantation, as well as you know about the autoimmune diseases. You know about the autoimmune diseases. What happened in the case of autoimmune diseases? Our own immune cells are directed against our own cells, normal cells. Okay, it means what? It means these MSC molecules are not longer functional. They are no longer functional in our body. That's why our cells not recognize that, okay, this is my own cell, I don't have to harm it, I have to protect it. But these MSC molecules, they are not present there, then our cell will not recognize it properly. okay, and destroy our own cell. That's the one concept related with the autoimmune diseases. Keep in mind, move ahead and understand another concept. You know the blood transfusion. In the blood transfusion, you understood that in the surface of the blood groups, there is a presence of antigen, antibody. Now I am telling you, in the antibody, okay, take, this is one antibody, okay, antibody is made up of two light chain and two heavy chain, okay, joined by the disulfide bond, okay. In this pocket, antigens will be fit and there is interaction between the antibody and antigen, okay. But this antibody is made functional. How it recognize that, okay, this is not my cell? These MSC molecules will tell. They go first, they bind with the antigens, okay, and make them we can say recognize to our antibody, okay, this is not our antibody, not our antigen, we will destroy it. So in the blood transfusion, this MSC molecules is also involved, okay. Now move ahead. That's the one aspect related with this. Now for our study, what we are studying is the immune system. Okay. In the immune system, first, till now you understood this thing, major histocompatibility complex, it's involved in the differentiation, is secreted by the chromosome number 6, which result into the formation of these molecules. Clear? Now they are mainly two types. We are calling as in the class form. They are a class 1 MSC, class MSC 1 and class MSC 2, okay, class MSC 1 and class MSC 2 molecules. Clear? Now you know that first bacterial toxins must bind with these MSC molecules. First MSC molecules must bind with these cells, okay. Now you know that there are two types of, mainly the 4 types of T cells, okay, but the 2 main types which are involved actively in destroying the microorganisms, that are called the CD4 cell, okay, and CD8 cell. CD8 cell are called the cytotoxic cells. CD8 cells are called the cytotoxic cell, okay. MSC class 1 molecules bind with the CD8. Remember, CD8, okay, that's called the cytotoxic cell, and class MSC2 molecules bind with the CD4. Let me repeat it again. There are 2 types of MSC cell, okay, MSC1 and MSC2. MSC1 molecules will go to the CD8 cell, cytotoxic cells, and tell them, okay, the things which I am carrying is not useful for our body, you destroy it. Or they are also saying, okay, what type of antigen I am bearing is useful for my body, you won't destroy it, okay. Same in class MSC2 molecules, they bind some of the toxins and they inform the CD4 cell that it is a foreign cell, okay. And you know, I already told you, the CD8 and CD4 cells, they are bearing the TCR, the T cell receptor, okay, and on that very receptor these MSC molecules are going. Understood? Now you understood once again that there is a major histocompatibility complex which is secreted by the chromosome number 6, which is mastered in the differentiating between the self and non-self. Clear? What happened? They are divided into the 2 types. The class MSC1 molecules go to inform the CD8 cell, whereas class MSC2 molecules goes to the CD4 cell. Understood? Now come again to understand the story how our T cell is functioning. Okay, how our T cell is functioning. Now we are at the point where we are discussing that this is our T cell, okay, which have the receptor, This is our T cell which have the receptors, okay, and there is now the MSC molecule. Suppose if this MSC molecules is a MSC molecules 1, okay, and bacteria secrete the foreign substance or antigen, okay, suppose this is bacterial antigens, it bind with the MSC molecules first, and these MSC molecules come to the receptor. This is the TCR receptor, okay, this is the T cell. Now we understood the whole story, okay. Now once it will stimulate, the MSC molecules stimulate our receptor, our T cell receptors, okay, and you know that MSC1 molecules is involved in activating which T cell? Very good, CD8 cell. Which cell? CD8 cell, means it is a CD8. It is a CD8, or also called as cytotoxic cell. Cytotoxic cell. They come, inform it, okay, this is not our what? This is our not desirable antigen. You destroy it. These T cells, they secreted some of the substances, chemical substances, okay, and by the help of those chemical substances, they regressively destroy this antigen. They regressively destroy the antigen, okay. That's the one function of T cell. That's the one function of T cell, and this function is called as the cell mediated immunity. This function is called, because they are produced by the cells, okay, that's why they are also called as the cell mediated immunity. So write down the first function. The first function of the T cell is cell mediated immunity. Hope you understood this. Cell mediated immunity. Clear? Now moving to the second function is the helper function. Second function is the helper functions. Remember, this MSC molecules is also called antigen presenting cell. MSC molecules is also called antigen presenting cell. Write down this word. This is very important, because you come across this word again and again. Antigen presenting cell. Now you are knowing why they are called presenting. You know, like they are taking and presenting in front of TCR. Take it, it's not mine. Take it, it's mine. Understood? That's why they are called the major antigen presenting cell. Even though macrophages are also one of the antigen presenting cells, but these MSC molecules, they are a major antigen presenting cell. Clear with this term? They are presenting the antigen into the T cell. Clear? Now moving ahead and understanding the second function. The second function is helper function. Second function is the helper functions. Now you quite familiar with the stability of these cells. The second function is the what? The second function is the helper function. HEL, helper function. You know, you have the helper cell, that's called the CD4. Why? Because the cell are helping by their 2 hands and 2 legs. So they are called the CD4 molecules or helper function cells. Suppose when they bind, these CD4 molecules bind, okay, they are not a cytotoxic cells, okay, they are not destroying directly the microbes. These CD4 cells, once they receive the antigen by the MSC molecules, they send a proper kind of stimulus, or we can say generate an immune response. Okay, you know what are the immune responses? Like the inflammation, fever, coughing, sneezing, wheezing, all are what? They all are the immune responses. Clear? So they generating the immune responses and activating the another powerful cell. What are the another powerful cell? The another type of lymphocyte, that's called the B lymphocyte. So they activate the B lymphocyte B lymphocyte, clear, and also activate their one on cousin brother. That cousin brother is called, very good, CD8 molecules, because they are the directly involved in killing the microbe. They are called the cytotoxic. They are not a, we can say, the useful for the cell, they are a toxic for the cells, because they are secreting some of the chemical substances which destroying the, discarding the microorganism. So they are called CD8. They are helping also the 8 molecules, clear. That's why they are called the helper function. Remember, first function is what? Cell mediated immunity. Second function is the helper function. Understood? Now move ahead, understand the another function. Another function of the T lymphocyte is the cytotoxic activity. We already understood, but go bit detailed to understand this term again. Cytotoxic activity. The third function, cytotoxic activity. Now I am going to ask the question, which cell done this process? Very good, the CD8 molecules. In context to the microorganism, not for us, not it us for is beneficial, because they are helping with their 2 real hand, 2 real leg, and 2 imaginary hand and 2 imaginary leg. Okay, that's why they are toxic for the antigens, because they are helping too much. Okay, but for us that's a useful, for us that's a very very useful, we can say. Okay, now these CD8 molecules, they are secreting some of the chemical substances, okay, and that chemical substances will make cell to die. They direct cell to die. That means programmed cell death. That's called the programmed cell death. And you know, there is one standard term to denote this kind of situation, that called the apoptosis. That's called the apoptosis. So apoptosis is a process by which a cell will die because of commanding by some of the cytotoxic cells and another cell also. Clear? That's the cytotoxic activity of T cell. We are in the third function. The first function is that cell mediated immunity. Second function is about helping the another cells to be active, and the third function is the cytotoxic activity. Now I am going to discuss the last function of our T cell. Till now we are not touching the B cell, okay, we are just at the point where we are discussing the T cell functions. Okay, the last function is the memory cell. Fourth function, memory cell, memory function. Now, do you heard about one term called the immunological memory? Okay, we have so many kind of memory where our beautiful experiences are stored, our, we can say, the achievements are stored, our pleasures are stored, our emotions, pain, everything is stored, which are disturbing us some of the time they are not functional, they are suppressed in our subconscious minds. Okay, but sometimes they become conscious by some kind of the stimulus. Okay, they are arises, they evoked again, they provoked again by the some kind of the stimulus. Same thing here also happened. I already told you initially that the T cells, when they went into the thymus, they become mature, differentiated into the 4 types. The first type we already discussed, cytotoxic cell. Second type we discussed about the CD4 molecules, also called the helper cell. Third cell is suppressive cell, and fourth cell is the memory cell. Okay, so this is a memory cell which recognize the previous encounter of pathogens. Okay, let me repeat it again. This is the one cell which tell the other T cells. What other T cells? CD4 molecules and CD8 molecules, that's the cytotoxic cells and helper cells that you previously encounter these cells by this kind of activity. So now you are just start this kind of activity and destroy it very fast way, very regressive way. Okay. So these memory cells have the knowledge about previous encounter. Previous encounter. Encounter of what? The pathogens. They have the memory about the previous encounter of the pathogen. That's why when you are vaccinating a person, when the vaccinations happen, in the vaccines, what actually happens? In the vaccines, there are dead attenuated cell antigens, okay, pathogen antigens. Suppose I am affected with polio virus, okay. In the polio virus, there is a one vaccine, polio vaccine, and there are also some oral, we can say the oral droplets are also discovered. So in the previously the polio vaccines, okay, in the polio vaccines contain what? They contain dead polio viruses, small amount. So when these dead amount is come to our body, okay, this dead amount will come to the body, those MSC molecules, they bind with those things because they are not our own, okay, and they are not so much potential that they come and they causes the poliomyelitis. No, they are the dead cell, okay, but still they have some kind of, we can say, the antigenicity, okay, they have the antigen producing power. So those antigens is taken by our T cell or cytotoxic cells, okay, and they destroy it because they are not alive, polioviruses. So once they destroy, suddenly the memory is generated and memory is stored. Now again, if the live poliovirus will come, we have prepared antibodies, autoimmunity, because these are also involved in activating where? The B cell, which is the actual fighter by the antibody. B cells fight with the antibodies, 5 classes of antibodies, okay, IgG, okay, IgA, IgM, IgE and IgD. These 5 classes of antibody, they are produced by what? They are also called as immunoglobulins. They are produced by the B lymphocyte, okay, and we understood one of the third function of this T cell is they are helping the B cells, okay, to recognize the coming pathogens, okay. So when the attenuated viruses, dead viruses comes to the T cells, T cell will destroy it, the memory is elicted, memory is generated, that's called the primary immune response, okay. That's why there is booster dose, 2 times doses again giving, okay. Previously memories are stored, then now again memory are so strong, they generate the secondary immune response, and that secondary response is so intensified that they are kept inside our body. So when we will expose again to the polioviruses, that poliovirus won't affect us because we have the memory cells, our cells are already prepared, they directly kill that thing. Clear? That's the basis, one of the basis of the vaccination is we have the memory cell, okay. Now you are thinking how beautifully God created our body. How beautifully God creates our body, inch and by inch we are precisely beautiful, wonderful, handsome, smart, as much as things you can tell about this. So by this we concluded today class. Is that first we will see the slide and then we conclude today our class. We understood the functions. First is the cell mediated immunity, okay, cell mediated immunity. It's a crucial for defending against intracellular pathogens such as viruses, certain bacteria and fungi. Then we have the helper function, stimulate the other immune cells such as the B cell and cytotoxic T cells to enhance their activity. Then we have the cytotoxic activity. Cytotoxic activity is done by mainly the T cell which kill or infect the abnormal cell by inducing apoptosis, programmed cell death in the target cells. Then we have the memory cell. Memory cells are formed during the initial immune response of can quickly respond to subsequent encounter with the same pathogen. So with this we concluded today class. We studied today mainly the basic review, okay, basic review about the T cell, okay, T cell lymphopoiesis, which is basically the differentiation maturation of T cells starting from the intrauterine life to the bone marrow house to the thymus gland. Then we studied the four different types of the T cells. Then we studied about the function of the T cells, and bit studied about the MSC molecules, which is very important for us. That's called the major histocompatibility complex. Okay, so thank you with having me. Bye, have a nice day.